they call it? Like when you make something out of any leftover in your refrigerator, it's like a
Call it a garbage salad.
Garbage salad, yeah, exactly. So pretty much it was a garbage salad, and it worked. I don't even really know how it worked, but it worked. And I went over to Mario's office, and I just knocked on the door, and I said, hey, I want to show you this. And so I just showed him a printout, and like he paused, and then he looked back at me. He's like, you just did the first 18 color experiment.
Welcome, everybody. My name is David Leclerc. I'm Technical Director here at the CAT facility at the University of Chicago. This is the podcast number 4 for the ChuG group. I'm here with Kelly Lunson and Pratip Chattopadhyay.
Not bad.
Not bad, thank you much. You know what's funny? I mispronounce my name sometimes it's easier to go through life when people are, Leclerc is not a common sound in English.
Yeah, I accept that people look at me, they say, how did I do? And I always give them, big thumbs up, right? They can massacre it, and it's,
I've been practicing since I met you, which is easily like 10 years ago.
Yeah, right.
And I think I have it right. It's Chattopadhyay.
No. I should have just, I'm going to give you the fake thumbs up, okay. But it's Chattopadhyay.
I'm not even close.
Yeah, that's why I said no.
Right? Whatever. It's like I said in the last episode that it's like being Cher. You just get to be Pratip.
Exactly. Yep.
Right.
All right. So I'm Kelly Lunston. I am an independent consultant with my own business doing consulting in cell-based analysis, assays and fluorophores and chemistry and such. And we actually already kind of introduced Pratip. But this is our guest, Pratip. He's at NYU currently, although a lot of us know him from NIH. That was about 2 years ago. 2 years that you
no, it's almost 4 years now.
4 years. Oh my God, I'm getting old. Yeah. So we are just very, very thankful for you to come and spend time with us. We had mentioned in the last episode that we really want to just build more community, get to know more people. Basically get to know people on more of a, not a personal level, but within cytometry, we are a tight community, and it's really good to know everyone and hear about your path in cytometry, which has been extremely eventful, and kind of, advice that you have for those of us going forward. So we're just very thankful for you to be here.
Thanks. No, I'm glad to be here, and it's always a pleasure seeing Kelly. It's great to meet David, and so thank you for the opportunity. So I guess I'll just give myself a little formal introduction. Even though you might see a weird caption at the bottom there, that's for an entrepreneurial workshop for NYU that wanted us to change our profile names, and I never changed it back. So anyways, I am not with something called TerraFlow right now, which doesn't exist, but instead am with NYU Langone Health in Midtown Manhattan, where I founded, I guess, and lead the precision immunology laboratory there. And we have kind of 2 roles. We have a research role, which is a typical academic research type role. And we also essentially have a core service that supports cancer clinical research and does some work in trials. And we also do work outside of cancer in autoimmunity and infectious disease. And basically anybody who comes to us with a cool sample set, we are happy to apply high parameter single cell technologies to it. So
that would include, do you do CyTOF still?
No, we don't do CyTOF.
Just single cell sequencing and then just super high parameter flow?
Yeah, exactly. And we've got, I shouldn't say this publicly, but I got stuck doing Luminex and stuff like that as well. But, which is cool. It's still high parameter stuff, but it's not single cell. And I love being around the cells. So, so we do some other things as well. It's for service purposes. Yeah.
Awesome. So why don't we just start out kind of from the beginning and work our way through your entire life and see how that goes. So how did you even get involved in immunology to begin with? Like, what was your progression to kind of decide that was what you wanted, and how that took you into flow cytometry?
Sure. So I actually was first interested in virology. I had an incredible virology professor in college. I went to the University of Virginia, and he just got me excited about studying viruses, and in particular HIV. And HIV at that time, this was like 1990 to 94, was a disease that had been around a long time, that was not really treatable. AZT wasn't very effective, and the cocktails that later came around, didn't really come around until 1996 or so. So the drugs that are common nowadays certainly weren't common then, and it was a death sentence for people. And I was fascinated by how essentially HIV took out the army that was against it, the immune system, right? And so so that got me really interested in virology. I did a bunch of research projects at UVA, and then came time to make a decision about what I was going to do next. And I decided to go to grad school. I was particularly enamored of the program at Hopkins because they had a school of public health, and I got my degree, my PhD there essentially. So I was around all these people who were working in public health, who were doing applied international work, epidemiology, stuff like that. And I was in the immunology and infectious disease department. And basically that turned into the molecular microbiology and immunology department while I was there. And that's where I got my PhD. I started doing HIV work. And the mentor that I had who was doing virology stuff, when I rotated through his lab, I was like, well, I'm not so interested in the virology. I'm just more interested in the immunology of this. And so luckily there was one professor who was moving into our department, and he was involved with the multi-center AIDS cohort study, the MAX. And that was a landmark study that started right at the start of the epidemic in 1983, 1984, and was tracking HIV positive people and just monitoring their immune response and doing virology work, and also doing important behavioral and sociology work essentially. And there were 4 centers. It was a really cool environment to work in, and so I started doing my PhD there, and I was studying why over the course of most of HIV infection, you lose CD4 T cells, but CD3 T cells kind of stay maintained. So the total T cell count in people is maintained for a long time in the face of the loss of CD4 T cells. And then all of a sudden, about a year and a half before AIDS diagnosis, the total T cell counts also drop. And it was a reliable marker for an upcoming AIDS diagnosis. And so my thesis was essentially to understand why that was happening. Was that some change in virus that wiped out a different subset of cells, or wiped out the production mechanisms of T cells, or were there other changes in T cell production that might affect that? And that got me really interested in the kind of nexus between public health and to a little extent technology, because we were doing new immunoassays and just the basic pathogenesis issues of HIV in the immune system. So
was that your intro to flow during that period? Were you doing kind of basic 6-color assays?
So yeah, even more basic than that.
Oh, really?
So 1995 or 96, I got started in flow, and we basically had a T cell panel. The T cell panel, compared to the 25, 30 parameter T cell panels we're doing now, this was a 3-parameter T cell panel that measured CD3, CD4, and CD8, and they ran it on every patient in the MAX. And I added Ki67 to that, and basically did the first intracellular 4-color experiment at Hopkins. And so which is a ridiculous achievement, but I'm proud of it to this day. Nobody gave me a certificate though.
Back then, the number of fluorophores available was like very limited. Like you had FITC, P, and
PerCP.
PerCP would be the last one.
Yeah. I think
APC.
Yeah,
there was still a red laser.
Okay.
Yeah.
And so, so we're running that on a Coulter instrument, and I was basically writing down numbers off the screen, and that was data analysis, right? And so it was all very rudimentary, but it was fun. It got me a degree. It got me some incredible experiences because I used to recruit my own patients for the study. So I would actually get to meet gay men who were HIV positive. That was one of the cohorts we had. Another cohort was IV drug users in inner city Baltimore. And, being a 25-year-old guy who just grew up in the suburbs, to have exposure to people and real exposure. I mean, I'd have real conversations with these people. They wanted to tell you about their life. That was a really neat experience. And, other than, doing the first 4-color flow at Hopkins, that is an experience that stuck with me, and I'm very lucky for it.
So I actually had no idea that there was a maintained level of CD3 positive T cells.
Yeah.
What, you just had an overabundance of one versus the other?
So basically, that was the question, right? Was it that CD4 and CD8 cells were being produced at the same rate, but then the CD4 cells were getting picked off, and you just had more loss of the CD4 cells, so almost like a fast churning of the system? Or was it that something would production of T cells that would basically, the bottom would fall out and you just wouldn't be able to make any more T cells? So is it increased destruction or reduced production? And the answer in my thesis was both. Basically, there was evidence for both. And so and we had hoped that we could understand whether there was a kind of rheostat in the body, almost a thermostat, to maintain CD3 T cell levels so stably. And we never really got to those experiments, but that's still an area where I think it's still an open research question, actually. Are there mechanisms that monitor how many of a particular subset you have in the body, homeostasis mechanisms, homeostatic mechanisms, that and are they T cell relevant, or are they memory cell relevant, or are they just immune cell relevant, or what? And there's probably been some work looking at homeostasis in memory cell populations, but not so much of the overall T cell compartment. So
I didn't realize that you had actually started straight into HIV/AIDS research to begin with, but it sounds more like you were doing immunomonitoring, not so much vaccine-centric work. So you went to VRC?
Yeah, so basically I was sort of. So basically I met my woman who later became my wife at the end of my PhD, essentially, and she was up in Philly. She was finishing up medical school, and so I needed something up in Philadelphia. But these damn doctors, when they're in training, they get matched wherever will take them. And so there was a period of 6 months before match where she was finishing up her medical degree, and I was I got hired as the first postdoc for Johnson & Johnson at a company called Centocor, and Centocor made an anti-TNF drug. I think it was infliximab or something like that, but I can't quite remember the name of it. But it was an antibody against TNF that they give to rheumatoid arthritis patients and stuff like that. And this was around 2000, so this is before like real immunotherapy. They hired me because they had some new drug in their pipeline for respiratory illnesses, and I was going to work on that. I was going to be a postdoc there, centering on that. I was very excited about it. I get there the first day, and the guy who hired me takes me into his office, and he says, so you're not going to be working on the anti-CD whatever project. And I was like, why? That's a really cool project. And he's like, because it kills monkeys. It killed every monkey in the preclinical trials. And so I was like, wow. So, and I didn't necessarily know the whole process. I was like, so you guys are going to test that drug? He's like, no, that program is just killed. And so I said, so yeah, I said, so what do you want me to work on? And he was like, well, pretty much anything you want. and we're transferring you to the clinical pharmacology department. And I was like, I don't know anything about clinical pharmacology. I don't even know what clinical pharmacology means, at that point, right? And so, so they transferred me to that department, and they kind of left me alone to do whatever I wanted. And so I go in and I go into their little library, and I'd, read journal articles, and I'd just be inventive, basically. And I came up with this idea for a chip that would be based on microarray type technology that would look at that would bind T cell receptors. So essentially, it would have MHC with different peptides across all the whole chip, and then you could see if there was. It would be a better way to see if there was a clonal T cell response in a sample, and it would essentially be using a microarray type platform. And so I started to, sort of build it and propose it really. And then I had some contacts with Affymetrix, and we started to kind of work on it together. And that's when Trishna matched to Georgetown, and she was going to move to Georgetown. I was like, well, I'm not all that into this project, and let me see if they'll maybe let me take it with them, just because I spent some time on it, and maybe it's worth something. And so, to their credit, they didn't claim IP or anything like that. It wasn't really in their wheelhouse anyway. So they're like, yeah, stick around to some postdoc mentors who worked on T cell receptor biology and stuff like that in the Baltimore and DC area, and they got no bites, because who wants this idiot working on his random project as a postdoc? You run a lab, you have projects that you need to get done. You don't need somebody sitting around there, doing some sci-fi stuff. And so I called. I happened to be in actually in my old lab at Hopkins, and I saw an ad come out on the cytometry list that Mario Roederer was looking for postdocs. And I was like, well, I'm here. I've heard of him. He sounds kind of cool. What happened? So I just cold called him basically, and said, I'm looking for a postdoc position. This is my background, and I'm in the area. Would you entertain me stopping by? He's like, yeah, absolutely. So I had a really nice conversation with him the next day in person. Came back and gave a talk, and it was love at first sight. I mean, I fell in love with the VRC. It was, I really thought Mario was going to be a great mentor, and he was. And it was awesome. I walked out of there. I still remember calling my wife from the garage saying, I think I got a job. And, and my parents she was like visiting my parents or something like that. So everybody on the phone was like, yay, it was great. It's a great moment, so.
That is so cool.
I remember a presentation you made. I don't know if it's at LIFCA or some Cyto event, but you were talking about the two-body curse. And I'm not sure if you coined the term, but I've been using it ever since. But essentially,
I certainly didn't coin it, but boy, I lived through it. That's for sure. So it's my wife is is really smart. Really, what's the word I'm looking for? Oh gosh, achievement oriented, driven, achievement oriented, she wasn't going to take a back seat to some idiot with a microarray. Or a flow cytometer or whatever, so during her training, it came up a lot of times. She's a hematologist oncologist, and so so she went through residency, through fellowship, and then her first academic stint, and then industry jobs. And it was difficult in a lot of ways because, nobody in academics gets offers for faculty positions from 30 places, right, that they can choose where they want to end up. And I remember I had gotten an interview at the Baylor Institute of Immunology, maybe sometime in the 2000s, I can't remember. And I had some kind of like bug or something, and I was so determined to go to the interview that I was like, I'm getting on this plane no matter what. And it was partly because, you know, it was the only gap in my wife's schedule where, you know, she could take care of the kid and I could be away. And I was determined to make it happen. And I remember they took us to a big French dinner, and, you know, and I was still like queasy from the day before, from the, you know, from that morning, basically, you know, having thrown up and stuff like that. And I was like, oh my God, now I have to drink and eat like, you know, French food, basically, for, you know, rich French food, elegant French food in front of these folks, and, you know, seem enthusiastic about it, you know. But it's a real challenge for people. And I don't know, there aren't, unfortunately, I want to be positive about it, but it's hard to be because there aren't a lot of solutions to that. You know, you hope that wherever you or your spouse lands up, the other spouse, the trailing spouse, they call them, gets a good position, and the university that you're going to will help them get that position. But the fact is that there are so few things that, you know, sort of fit the right, you know, all the check off enough of the boxes that it is really hard to pull off. And so when we moved up here, you know, she said, well, you know, it's kind of your turn. And that's how we all looked at it, that, you know, any move was either we stay in the area and so on. And anyways, she was looking to leave her position. She'd been there for a few years and wanted to try something new, and was thinking that she'd end up working in Boston. There's enough biotech here in New York City that it's okay. So we managed the 2-career problem. And it gets better for anyone who's going through it, you know, as mid-career or a young career person. First of all, sorry, you know, just look at your spouse and remember that you actually love them. And, you know, there's a whole reason to go through this. It's really tough. It's really stressful. But the nice thing is that a lot of that's just front loaded early in your career. And then later on, you start to develop a certain maturity about, you know, what's important in your life. And also sort of that pressure and the possibilities also get sort of wider. And so it does, that 2-couple problem kind of does get relieved after a little while.
And the kid grows up, they get a little bit more mature, a little bit more self-sufficient. They can handle you being gone multiple nights of a week. You know, those sorts of things are always difficult too for anybody that travels for their job.
Yep.
So that led you to NIH. That led you to your marriage with Mario.
And I loved it. And, you know, the first, Mario's very, he's a great mentor in that way. He doesn't stand over your shoulder and, you know, and keep track of what you're doing every second. He proposes the big picture and, you know, gives you some guidance, and then it's, you know, it's up to you to go for it. And
what was that first project at NIH? I mean, so I always sort of wondered, like, it was, you know, QDOT's enabled the 18 parameter flow stuff. Were there other people besides you who were kind of fast on that trail, though, to validate these assays and mainstream that level of cytometry? Or were you guys just kind of acting on your own, being like, we're willing to take on this burden of QDOTs to accomplish this goal?
No, it was mostly, this is probably a better question for Mario, but because that was right when I was coming into the lab. And I actually remember Marcel Bruches, who was the founder of the QDOT Corporation. And he was, one of my first memories in Mario's lab was him sitting at the edge of the bench, you know, pipetting out stuff. I was like, who's the student? What's all that colorful stuff?
Well, that makes sense. Marcel did all your conjugations? Because
well, he didn't do all the conjugation. I did the conjugation. But he prepared all the dyes, which was not trivial. I mean, you know, and you know this probably better than I, Kelly, but you know, the QDOTs are not easy to make, essentially, you know, and the conjugation process was not that bad, actually. It fit the way we did PE, and, you know, and
it's the purification, not the conjugation.
Yeah, exactly. So, you know, so the purification would be, Well, so we would get reasonably purified material, and then after the conjugation, we just went through the same purification steps that we do for PE and all that, and it seemed fine, at least for the surface markers. We never had it working intracellular, but for the surface markers, it was okay. But you know, so the first project I had was a crazy thing where give up Mario's big idea or something like that. But we never got it to work. I don't know if he ever did it after I left, but it was like this self-assembling to essentially use oligos to self-assemble with each other and create some kind of, you know, ongoing reaction. I can't remember all the details.
Fluorescently, like for tagging antibodies or something like that?
Oligos, basically.
I mean, it sounds like Codex, so.
Yeah, it's not that far from what they're doing with Codex. Yeah, and so it might have been along those lines, but I never got it to anywhere near working. And so I quickly, you know, not cozied up to Marcel because I didn't really know him at that point, but we had these Q-dot conjugates in the lab, and I started just building T-cell panels with the conjugates, and it was just a kind of a struggle. They were just testing things sort of randomly, and they were in the refrigerator, and so I was just staining PBMCs, you know, kind of getting panel learning, iteratively learning panel design kind of on my own. And then I mixed them with the other, you know, with the other channels. And one afternoon, I just tried like an 18 color experiment, basically, that, you know, we had like one, you know, reagent in every channel was what was available.
About it, I'm like, that sounds really well thought out. That was really well planned.
It was pretty much whatever the, you know, it's, what do they call it? Like when you make something out of leftover, any leftover in your refrigerator, you know, it's like a
garbage salad.
Garbage salad, yeah, exactly. So pretty much it was a garbage salad, and it worked. I don't even really know how it worked, but it worked. And I went over to Mario's office, and I just knocked on the door, and I said, you know, hey, I want to show you this. And so I just showed him a printout, and like he paused, and then he looked back at me. He's like, you just did the first 18 color experiment.
Do you feel like you became famous after that? People would be like totally in awe of you and be like, whoa, that's pretty.
I had no idea, but it's, you know, I paid those people handsomely. So, you know, so yeah, so that was how the first 18 parameter experiment came about. And then somebody, a couple of, they were really advanced postdocs there. A couple of scientists, David Price and Mike Betts, were in the immunology labs downstairs. And David's particular you know, expertise was in making tetramers. And so we started to make tetramers with quantum dots. And then Mike helped with some panel design and things like that. And that's how we basically put together the first panels in that paper, along with Steve DeRosa, and who taught me everything in Mario's lab, basically how to stain and in conjugations and all of that. So you know, in a lot of ways, I owe my career to Steve DeRosa and Steve Perfetto, who taught me how to use the instruments as much as, you know, to Mario. And so so yeah, so that's, you know, that's how it came about. And, you know, back then you could do cool technology and, you know, get it into Nature Medicine. Since then, I think it's gotten a little bit harder, although, you know, nowadays there's so much cool technology that you see.
Now you just need 40 colors to get a paper.
Yeah, exactly. And you're lucky if you can get that, right? And so, you know, I thought with the brilliant dyes, that would be, you know, a high impact paper, and that might be a Nature Methods paper. And you know, that wasn't interesting enough for them, but they've had such a impact on the cytometry community, right, and on immunology in general. So, you know, it just goes to show you that you shouldn't measure the mark of you know, your work by the publication that it gets published in. You know?
Yeah, absolutely. So I don't know if it was one of the first times I met you. I started, you know, attending some of these CHAVI, or CHAIVI, right? Because CHAVI in the UK actually means like a bloke or something.
Oh, really?
Yeah. They would like laugh at me all the time when I would give talks about the CHAVI. But, you know, I knew nothing about immunology, you know, when I first started at BioLegend. And that because we were just launching the Brilliant Violets, I was starting to attend a lot of these, larger meetings where it was also focused on HIV vaccines. Okay, I've also been absolutely listening to this.
That just reminded me that actually I think it was at a Cyto meeting that you were with, oh my God, why am I blanking on Brent? Yeah, what's Brent's last name? Why can't Gaylord. Gaylord, right, with Brent Gaylord, and you were like
introducing him to everybody that year.
Yeah, and you introduced.
They didn't really, they had been so completely interested in it only as a chemistry. The first molecule that they had brought to us was so ridiculously big and cumbersome, but it had these amazing optical characteristics, and we're like, that is not useful for biology. And they had been so focused on nucleic acid detection and things like that, which was not our interest. It was really, so bringing them to some cytometry meetings so they could see how people were applying Q dots or what the limitations were for, optical techniques, was a really, monumentous thing. It's why I introduced,
Yeah, and I mean, that was great. I'm still like, in reasonably frequent contact with Brent, and that's.
Yeah, where's the brilliant yellow greens? That's what I'd like to know.
Yeah, well, let's get him on one of these podcasts. Right, he'll be cagey. He's sharp. He can.
Yeah, we won't get any information. It'll just be a lot of hemming and hawing.
Right. So, but that was another one of those, like, let's just try it. And so, so the consequence of that meeting was Brent just sending us some, conjugatable material or something conjugated probably already.
It was ours. We were making.
Oh yeah, you were making.
Right? Yeah, we had wanted you guys to be one of our first beta testers.
And so I tested that kind of on the down low. I didn't tell Mario about it, and I just tested it. Right, it's just coming in the mail. So I just tested it without. It was probably the 421. I compared it against Pac Blue or something like that, and I took that was another one of those times. It took the data to Mario, and just went into his office with the printout, and I said, this is a new dye that that Kelly and Brent sent, and what do you think? And it was phenomenal, right? It was like super bright compared to the Pac Blue, which was really dim, and he was like, wow, holy cow. And so that was the start of the interest from the VRC.
I think when it came to the publication on that, it wasn't its application of flow cytometry that was the science-worthy paper. It was the entire optical tunability of a polymeric fluor for cell biology applications. If we had the time to kind of widen the application a bit, like now that I think back to that first year and the kind of work that we did around it, it was so limited. It was so not sexy, and there was so much more that we could have done, up front that I would know to do now. But we had never done anything like that before. No. So just getting it into people's hands, being like, can you break this? Is this gonna completely fall apart? Like, what? Right.
I mean, and it's not like we were turning around looking at new dyes every 5 minutes back then.
Not like we are now.
Yeah, exactly. So yeah, it was a very different time.
So I still, to this day, tell my friends about these CHAVI meetings, though, because I personally learned so much about vaccine development, all the different viral vectors that they were looking at using to deliver different kinds of vaccines. I know this is like going to be the dumbest, most simplest question, but why don't we have, like, what are the fundamental issues? Is it rate of mutation of the HIV? Is it some inherent nature of the retrovirus that, like, we've definitely gotten to the point of being able to manage symptoms, but are we ever going to look at a future with an HIV vaccine, do you think, in your opinion?
Think so. I'm maybe more hopeful than when I was at the VRC early on. During my time there were kind of I guess phases of the way science thought, leadership thought about how to design an HIV vaccine. And early on, there was a great interest in T-cell immunity, that you would need a vaccine that elicited T-cell immunity, and characterizing what types of T-cell responses would be important and things like that. And that's really how I got, interested in T cells. And that's really where a lot of the panels that we built came from. And, it did a lot, that sort of phase of HIV vaccine development, or that era or period, I think influenced greatly what happens in immunotherapy in cancer now, because, a lot of the immunoassays have transferred there, and, a lot of the understanding of T cell immunology, comes from things that happened during that time. But in the end, vaccines, that T cell responses, it was deemed, these efforts that were geared towards developing robust T cell responses weren't actually panning out as efficacious vaccines. And so, what did really make a difference was the emergence of structural biology and the ability to characterize the viral proteins and the antibodies in particular that were neutralizing, and understanding, with just incredible resolution and precision how those bind, and and sort of re-engineering that. That is the direction that HIV vaccinology is in now, the era that it's in now. I remember when I was at the VRC, it was almost like there were two camps. There were the T cell people and the immunology people, and then there were the structural biologists. And we'd all present to each other, but almost talk all over each other's heads. But I feel really fortunate that I was there during those early times, that where I got to learn at least a little bit about that, because that is actually one of that area has grown into such a successful, potentially successful path for vaccine development. I think that's where you're going to see the biggest impact on HIV vaccine development. So I don't think it's over yet. I don't think that we can declare it that we'll never get an HIV vaccine in our lifetimes. I don't think we can say that. But having said that, moving out of the field now for 4 years, I don't know where they're at, and so it's it's a little bit harder to give you an updated view. But
I've been listening to this podcast all through COVID, because I thought it was funny how you started this podcast talking about the differences between virology and immunology. I'm relatively new to immunology, and I had no idea virology was just so different. It's more like, this is the life of the virus, this is the effect on your immune system. These are two very separate things. And so This Week in Virology is like this amazing podcast that teaches me something every day. I swear to God, the last one I listened to twice. And they do a lot of referring to the non-neutralizing antibody production for HIV. So to me, when you're talking about the structural biology elements of it, sounds to me like, because you're not having a natural neutralizing response, maybe the solution is actually to engineer one.
Exactly.
Okay.
That's where vaccinology is headed.
Yeah. And it does, it also, I completely agree with you. You can see the transference of knowledge from the HIV vaccine effort into the almost more attainable, which seems insane, but more attainable goal of cancer vaccines. So that's, you're not in necessarily, your current research area is not in cancer vaccines, it's more in biomarker detection and diagnostic capabilities, right?
Yep. And
do you do extracellular vesicle work, or is it mostly bloodborne or blood?
Mostly blood, because I wanted to, so we look in the tumor. Actually, David, did you have a question though? I thought.
Just to clarify, the podcast we're talking about, you have a podcast as well? Or which podcast is?
No, I don't have a podcast.
No, it's called This Week in Virology. They're on their like 700th episode. It's just that in this last year, they've been dedicated only to COVID. So they're the only piece of news that hasn't caused me stress this year. Like even listening, I stopped listening to the New York Times because I couldn't handle it. But theirs is just data. It's just presentations, and it's pretty awesome. Anyways, keep going, Pratip.
Yeah. So I when I started thinking about leaving NIH, one of the reasons to leave was that that technology development was really cool. It was
like
it wasn't enough for me, and Mario's lab certainly was not just about the technology development. I mean, he made huge contributions in understanding HIV and animal models of HIV and other diseases as well. But you know, I was really on the technology side, and I felt like I was giving away these technologies for other people to apply. So so I wanted to be more application oriented and less
And you
tech.
have an inner entrepreneur too, let's be honest.
Yeah,
Like
yeah.
kind of always seeing opportunities.
Yep. And I, you know, and I did feel like I had been working in HIV for a long time, and that, you know, it would be great to try to make a, you know, a contribution in the cancer immunotherapy setting. And so so I, it was a really lucky and natural fit, I think, here at NYU to you know, to create this lab. And one of the things that we've been, you know, that I've been actively avoiding was, you know, anything that required a lot of tech development, because I knew it would be fun and I would like doing it, and I would, you know, it would be great, but ultimately it would put me basically, you know, back in time, right? You know, and so so I really focused heavily on trying to make 30 parameter flow, which, you know, which, you know, is another story of, you know, how we first did those experiments. We were under the gun to do those before Cyto, you know, to get the whole system working, but the hardware wasn't quite up to snuff, and, you know, there wasn't firmware for it, and, you know, it was a crazy mess. And, you know, it went in one weekend, the air conditioning had failed at the VRC, and I did all the staining, you know, over by the freight elevator where there was a little bit cooler, and you know, Mario was there with me for part of the day, and, you know, ran the samples. And as soon as you know, as soon as like a comp tube would come off, he'd look at it, he'd say, no, that's not good enough, you know, stain it again and put it on there. And then you know, I looked over his shoulder as we were sort of analyzing it together, and we were like, all right, 30 parameter flow worked. And, you know, he sent an email to the Purdue list. And anyway, so what was I trying to start talking about that? But the point was that I didn't want to be in that kind of tech development mode. And I saw that we needed to have better application of high parameter flow, right? Mm-hmm. And at the time, especially in 2016, 2017, there was a mad race of just new technologies coming out. And you know, I kind of felt like at that time, and feel maybe even more so now, that we have the technologies to do really cool things. Maybe not everything, you know, and there's always, you know, you never should say that tech development is dead or that you don't need any better technology, but gosh, we can do, you know, a whole lot with what we have. And so
sometimes I feel like technology development is like a tree when it sprouts new growth and then it blooms off that new growth. Like you have to give everything that's growing are in these stages. Yep. I've been doing a lot of gardening lately, but you know, when we have these big bloom or these, you know, these big growths of technology, we have to stop and ask ourselves, how is this best used? We now have these bigger and wider nets. How big do the holes of those nets need to be? What are, you know, how does it need to be woven? You know, like what kind of controls do we need to make sure that we're employing? What sort of statistical analysis, you know, control do we need to, you know, to make more prominent? Because that was actually one of my questions for you. I totally feel you on, as much as you so obviously get excited with technology, you can see your entire face light up when you talk about it. Yeah. It also pigeonholes us professionally to only be tool providers, when that isn't how any of us really are. We all have passions for, you know, different biological applications, and you are uniquely positioned through your pedigree and through your creativity and just who you are and your experience to be able to be one of those people who thoroughly understands all their tools, wants to hold the community to a certain expectation of how it's applied, and be able to do it yourself. Exactly. Because most of us that understand the technologies we aren't able to integrate it into the full picture for our careers.
Yeah. And if we don't have opportunities to apply it and take the time to really apply it, then, you know, development kind of gets stunted, right? Because then you're developing things just because they sort of pop into your head, but you're not developing things that are targeted to the need that is out there, the need for the application, right? And so, so early on when I came to NYU, the first thing I said is, people are going to want me to make new panels, and I don't want to have to spend time doing that and, testing every single panel new and, doing that off the top of my head. So we developed Color Wheel, which is a panel design tool that's based on spillover spreading matrices. And that really revolutionized our panel development for us. So people would come to us with lists of markers. They'd want to use it on our instrument. So we'd have the spillover spreading matrix for our instrument, and we'd be able to build these panels that worked the first or the second time.
So did it also take into account antigen density and things like that?
Yes. So we have the users come to us, the investigators come to us with a ranking of there she is.
Yeah, there she is.
With a ranking of density, essentially. Or if they want, they can rank by if they think that a lot of stuff is the same density, but this particular marker is more important.
You made an intelligent panel design tool.
Exactly.
Like what BD's been talking about for like the last 3 years or 4 years.
And then we sold it to BD. And so.
Shut up. Is that what Bob Balderas gives his talks on?
No. Bob's, well, yes and no. So Bob did a whole antigen density.
Yeah.
I wouldn't want to call it an exercise. It's almost a journey because.
No, it's humongous. And they're not the only ones either. I would say that's going to be one of the most important tools that we're going to have.
Exactly. It's incredibly valuable. And so so that's, and that can all be that's stuff that, I have in my head, or you Google from, or you just take a wild guess. Best way of titration, right? Look at it from titration. So we rely on our investigators to come to us with that information. And then we actually match that up against the spillover spreading matrix and choose the best channels for the dimmest markers, ones with lowest antigen expression, or the ones that are most interesting. And it's key because it's based on the matrix for your instrument. It's keyed to your particular instrument. So it's almost like it's guaranteed to work. It's not, it's guaranteed to work on your instrument. And that's the experience that we've had. So we've been building panels like crazy. They always work on the first run, second run at the worst. And it's great. It's a real testament to how things have changed. When I was in Mario's lab, we would build a panel and it would take us, a year, 2 years just to make sure that it worked and, and iterate to the point that it would work. And so, so our color wheel has been, it's somewhat been adapted. So, it's not quite specific to your own instrument now. And, BD did some other things with it, but it's part of their research cloud software that they'll have, or,
to me, this is kind of how I see our space right now. If we're talking about what probably can occur, is to have a tool that takes away a lot of the guesswork, the way that we used to have to do it. So, the antigen density tools that look at stimulation, disease state, all of and really just databases, how diseases affect antigen density, for just as an example. And then you have your spreading spillover matrix, you understand how your machine works relatively well, you can input those as a data set into something that then starts to make these intelligent choices for you. And that takes away, hopefully, so much variability and consistency in just the assays that we're even applying to the biology. But on the flip side, we just had our last podcast about software. And, like we had Jeff Cracker on because he worked at Cytobank, and also David Novo, obviously, he knows everything about, but the way that I see it, and actually is one of the best talks that I know that you've given a talk on it. I didn't see your talk at MetroFlow, but my friend here, Brandon Sells, he's at Pfizer. He's just raves about your MetroFlow talk, which is the correlation between our molecular markers, our patient data, being able to kind of cluster together more features than just the 16 parameters in the assay, but you're able to say there was a certain severity of disease manifestation, and there was this backdrop of these particular epigenetic markers, or I don't even know, or this person was a COVID survivor, and that becomes a data point. But there has to be a point where we are merging our molecular, data with the higher level phenotype.
Yeah, absolutely. So, so there's a lot in that excites me. The first is that just from the perspective of higher level phenotype, what we do with immunophenotyping is the amount of data that we generate and the depth of that data is amazing. And we oftentimes only scratch the surface of that data. And that's a shame, because we work so hard to, this whole history of developing fluorochromes and, and making all of these systems work. And then we just, pick out, all right, we only want to look at naive T cells or central memory T cells, but we have this depth of data that's underneath that is sort of undefinable cell types or not defined cell types. And one of the things that we've really pushed in, in these last 3 or 4 years that we've been around is to mine that data as completely as possible. And I've been frustrated that, you do a Tisney plot and you get a few islands and you compare those islands across groups, but there's not a lot of statistics associated with that. Of course, you can go back and look to see what cell types differ and then quantify those and all that, but it's a lot of work. And ultimately with dimension reduction, you are reducing high dimensional data, right? I mean, just by it's in the term dimension reduction. And so we've been taking an approach with our data analysis where we don't do dimension reduction. And we just let high-powered computing take care of analyzing every single type of cell that's there, every single possible phenotype that exists. And so we did that with some work in melanoma and have developed that. And then we kind of realized that was overkill, that, we were comparing patients on the basis of cell types that, were found 2 or 3 events across each patient, and there wasn't really much point to doing that. So, so Dan Freeman in my lab was a young computational scientist. He's got a really interesting backstory, but we probably don't have time for it. He was going to go to med school, did a summer with me, and then did really well on his MCATs, but decided it wasn't for him. Came back to work with me.
Won him over.
Yeah, I won him over.
Personality of science. Way to go, Pratip.
It was awesome. It's a loss for modern medicine, but it's a win for science or for researchers. And so, so Dan developed and sort of alternate approach that doesn't do dimension reduction, but makes all of these calculations manageable and allows us to analyze a huge number of cell types across patient groups. And it works really nicely, and we're building these simplified platforms to be able to analyze data and compare across patient groups. So when
should we be able to see publications for this, just as an example?
Sorry?
For this work, is this something that you're close to publishing?
Close. So we sent it to, this is another one of those, don't judge a quality work by, where it gets published. We sent it to Nature Methods, and they said, well, with any data analysis approach, we now require that you have an independent validation experiment. So it wasn't enough that we had 60, 70 patients and we had training set and test set. We need an independent experiment on a new cohort of patients to validate it, which is probably for the best and the right thing to do for science and all of that, but come on. So we just did that experiment now. So we're getting closer to having it published, and it's the method seems to be validating in other settings. So so I feel good about it. But the point was not necessarily to do the advertisement, but still has great value here. But the point was more that I think we need to think sort of on multiple levels, right? We need to think about looking at the data in as complex a way as we can, so that we're mining it completely, integrating it with other data types, as comprehensively as possible, and then stepping back and saying, wait, do we really need to do all this? Because ultimately, a 30 marker gene signature that comes out of a site-seq experiment, gene and protein signature that comes out of a site-seq experiment, is not a clinically relevant thing, right? Nobody's going to come to the doctor and say, hey, can you, I think I'm not feeling well. Can you give me, do some site-seq on me, right? And the doctor is going to go, yeah, I'm going to do that right now. That's not a really likely biomarker that, has validity. So one of the things that we like to think about also is doing all this work in that high dimensional space, but then boiling it down to as simple a potential biomarker as we can that still shows the difference between the patient groups, maybe doesn't tell us as much about the mechanism of what's happening, but is easy to measure and can be translatable.
That's really kind of what we joke about when we think about the history of flow, where it was like, well, it was 4 colors. Well, that's not a big net, right? That's like a net the size of my hand.
Yeah.
So
right.
you don't really get to call that, high content. Then we get to be like 18 colors, but then we get to be 40 colors. It isn't about the colors, it's about the content. It makes our net wider and wider in order to possibly gather in something more so that a more rare fish that maybe we hadn't seen before is more likely to be caught, so that then we can reduce it to what are the definitive markers that are necessary to actually give us information about that rare fish, right? Like that's really, as much as we get all tangled up in the details of the technology, it's because it's enabling the larger picture to become more complete in order that we can pull out possibly the more interesting, meaningful piece of that data.
Exactly. And, in our community, we tend to have, we are all nitpickers, right? we're all detail oriented. We're all careful. Cytometrists are incredible at 2 things, detail and education. I think, we love to teach and we love to get into the gory details of things. And I think it's valuable also for us to take a step back and see how we fit into the larger sort of biomedical ecosystem and, and what our role is. And we can all do that. I mean, that's not just for the people who are in, an academic hospital or in an industry setting where they're working with a lot of people from different environments. We can all do that, and there's incredible value to that. On the flip side, it's important for scientific leadership at institutions and things like that to realize that just because a new instrument comes along, that doesn't mean that it's going to necessarily bring a new value, and to talk to us as cytometrists about what instruments are needed and necessary so they don't go on shopping sprees that ultimately take away from doing, clinical research projects that could have a greater impact, right?
I've always said the machine should not be the thing determining the research. The research is the thing determining the machine. Like if you don't know what you're going to do with it, don't go shopping yet because you're not ready.
Exactly. And I see that, unfortunately, and that's, it's
so
one
I want to redirect the conversation just a little bit. What, like in this past year, what in your kind of application area, because you do have this, this link to vaccine research, what has the whole COVID pandemic taught you or instructed you most professionally? I know that you're kind of in the heart of it in New York. And what do you think you're going to carry with you past this year?
Yeah, so it's a great question. We had a pop-up morgue at the, pretty much, within view of our lab early on in the epidemic. And so you can imagine that it's, it's scary. And just from like, just from personal perspective, you don't think about being a leader of a lab. You don't think about asking people to come to lab and worried that you're actually risking their lives in some way, right? And that their families are worried about that. And I remember my lab manager, Jason, saying, right around the time when everything was shutting down and there were, decisions being made, but they hadn't been communicated yet. He's like, my family's really freaking out about me going to the hospital basically, and working every day. And you know, I don't think I can come in anymore. They're just really freaking out, you know. And that conversation will stick with me because it was a time where I had to make a decision. You know, I had to say, all right, is that, you know, is that going to be okay? You know, because essentially I'm letting a guy who needs to be in the lab to do his work, not work in the lab to do his work. And, you know, but from a personal perspective, like, yeah, I totally get it. You know, my family would be freaking out too. And so, you know, how do you handle that, you know, as sort of a young, you know, I managed people before, I guess, but, you know, it wasn't my lab. It wasn't, you know. And so, so what tone do you want to set and who are you? And I finally decided, well, I just have to think about this from like, you know, what is my gut reaction? What is the Pratip instinct? And the Pratip instinct was, don't come in. You know, if you're worried about it, don't come in. And, you know, if your family's worried about it, don't come in. We will figure it out. Let's just let things settle down for a few days. And
did your lab become involved with COVID research at all during that period?
So yeah, so everybody basically freaked out, including me, and we all stayed home. The mandate at the hospital from NYU was basically that, you know, for some period of time, it was that, you know, you could only be there if you really had to be there. And we were doing blood processing and staining for some clinical trials, things like that. But nobody's coming in for a cancer patient visit in the middle of that mess. And so all that got canceled anyways. So we all started working from home. And one of the pathologists called me up and he said, his name's Chris Park. And he said, you know, they're not really collecting samples from COVID patients because they're just too overwhelmed. And yeah, and so we had this sort of great pool of research material, but they couldn't get their, it was chaos. It was just hard to, you know, make it all work. So they were collecting like cell pellets that, you know, that we couldn't use for staining, right? That they could use for DNA analysis or whatever. And that was basically as much as they were willing to do at the time. So Chris said, you know, I've got my hematopathologist, he ran the training program there. And he said they have to be there anyways, because, you know, it's part of their job and they might be needed, but they have a lot of extra time on their hands because they're not reading slides from, you know, patients and stuff like that. So he goes, well, so do you think that they could stain COVID samples if we found some samples? And I said, well, yeah, I mean, we can figure out how to train them up. And I think I can, you know, I can teach them. And so, so basically what they did was they trolled around the emergency room and in the laboratories that were doing the diagnostic testing, and they just brought us 1 day old, 2 day old blood samples, whatever they could get, and then we trained up a second team of them to do my high parameter flow stuff, to run 2 or 3 of our panels, and one of them had flow experience before, a couple of them hadn't had any flow experience. Taught him how to do the staining, you know, set up the instrument, run the instrument, and get the data basically. And I just did all that training via Zoom, and we just left a little, you know, cell phone on the bench basically while they were doing experiments, so that they could just, you know, easily turn to the phone and say, Pratip, I don't know what I'm doing here, or, you know, I have this question. And the data looked beautiful. Data looked perfect. It was incredible. It's a real testament to how thoughtful they were about doing the work. And so we ran samples from 30 or 40 COVID patients who ranged the gamut of disease. They all went to the ER, so they were all typically, you know, at least moderate disease. But some of them ended up severe in the ICU. Some of them had short hospital stays, or some of them went home. And we found cell populations that were defined by checkpoint molecules, or molecules that are expressed on myeloid cells in our panels that correlated with outcomes. And so it was a real eye-opener to me into the fact that we could almost do cytometry remotely. I could teach anyone to do this. And, and that had been my ethos all along. When I first came to NYU, people were like, oh, well, who are you going to hire who's famous in flow, who's going to work with you, who's got a ton of experience. And I was like, actually, I'm just going to get people off the street. I mean, so I got people who had lab experience, but were young and, and basically
Teachable.
trained them up quickly. Yeah. And so during the COVID period, that was like a, sort of compressed version of that. And it speaks to the quality of the instruments and reagents that are out there now. And, we probably don't thank our vendors enough, but the development that they've made and the work that they do, the quality of it, reflects in the fact that any idiot can stain cells now, almost, right? And we've come to the point where we can democratize these technologies, which is really great.
Do you think that you will continue to put energy and focus on COVID-19 research going forward, or is this more of a purpose driven?
Yeah, it was that we had an opportunity there. We didn't want to waste it. I was reticent about, there's a mad rush to doing COVID research early on, and probably still is. And I was reticent about joining that rush, figuring that I already had a mission of what I wanted to do here. And, I just wanted to keep going with that. So yeah, I don't see us, I see us in a supporting role. I mean, NYU got a vaccine trial unit, and I'm theoretically research lab operations director of it. But the reality is that they're recruiting, good people who work in vaccinology full time, and so I'm sure my role will, just be advisory.
For you, the future is biomarkers.
Yeah, and it's.
So what this company, Terra, what's it going to be called? Terra.
Well, so we don't have a company yet. Let's not talk about
Oh, okay.
But one of the great things about being at a in an academic institution at a place like NYU, they really support entrepreneurship, and I'm not like you. I haven't had all this business experience over time, and and so understanding that ecosystem was new to me. So I've been trying to learn about that, especially now when I have less of a commute because I'm working from home a lot. I've spent actually a fair bit of time in their entrepreneurial setting, learning about that. And so our data analysis tool is TerraFlow. That's the one we're writing up now. And, and so we just imagined that
beta tester
as a venture. Yeah, absolutely. Yes. Well, we'll be back in touch about that, but we'll see what happens with that stuff. We just want to make sure that we can get the tools that we're developing out there in as impactful a way as possible, so that everyone can use them easily and they're not just kind of lost in the literature. That's the whole goal of all this stuff.
That's awesome. It really captures my imagination how any of us can be entrepreneurial when we are so detail oriented and so dorky about such a strong focus on where we see our discipline going. Yeah. And yet our brains in the background are like, there's these opportunities. How do we, with no experience in business, or never really having ever learned how to promote ourselves well, how do we manifest this entrepreneurial spirit? And it just is like an element of human nature. I find it very engaging.
It's really, it's interesting. You think about how our careers have progressed, and and I remember a time when I was frustrated coming out of college, thinking that I don't have enough ideas about science. Am I really going to make it as a scientist? And I should be more creative and stuff like that. And now I recognize that me and everyone else, we all have a ton of ideas, and and then it becomes a problem of execution, right? You know, how do you get your ideas actually, you know, realized and such, and you know, and having the time to do everything that you could possibly do and want to do. That growth that each of us experiences is something really interesting and cool to me.
Yeah, me too. It's very, what are our strengths? What are our weaknesses? They're also different, you know, between person. Yeah. And how do we manifest our strengths into these ideas that we have, basically. So this actually, I don't know if we achieved our goal at an hour or an hour and a half worth of a podcast. I think that you're so interesting. I would listen for an hour and a half, Pratip. I mean, you're just enrapturing to me. So, so maybe we should.
Hour and a half too. So.
Oh, stop. But it is. I think this does kind of bring me to what my last question was, which is, when I was being raised up in science, you know, which was actually neuroscience back then. You know, you're constantly acquiring knowledge and you're trying to do your best to make correlations between things and actually have original thought, right? That's really what they're training us to do for that first part of our careers. And then there's this second part of our careers, which is significantly more challenging, to be able to find good mentors that help us do something with those ideas and help us position ourselves professionally, help us sell ourselves and know what the next steps are and have confidence in ourselves, is, to me, has been significantly more challenging than the beginning of my career ever was. Yeah. So I'm definitely curious, with your experience and now that you are responsible for training up that next generation, like what your thoughts are on success in both the beginning and the mid. You're not quite at the end of your career, so I'm not sure if you can give us any sort of advice about that, but you can definitely give advice for the beginning and the middle.
Did the dean tell you something?
I know, like, you are not that old.
Exactly. You know, I think the last 3 or 4 years has been really fun for me in a way that I wasn't expecting, but I'm really glad. Surrounds the training of others, and you know, and just, you know, giving advice and thinking about how I'm thinking about doing things. And you know, I don't want to hold myself up as you know, or say that my advice is particularly valuable, because you know, in the end, I'm not some kind of crazy high-powered researcher, and you know, and I might not be as successful as other people, but one of the things that I tried to live by the first couple of years, and it's kind of fallen by the wayside, and I'm trying to reinvigorate it, is that I wanted to spend 60 to 70% of my time doing things that I just wanted to do, and that were just tasks that were interesting to me and relevant to me, and that I was driven by. And then another, you know, whatever, 15% of my time doing things that had to be done because they had to be done. And then another 15% of my time addressing like nagging, like really annoying administrative things or nagging things that I was going to get in trouble if I didn't do. And so I kind of tried to live by this ethos that 60% of my time plus should be stuff that I'm really excited about and working on that. And for the first couple of years here, it was definitely that. And I was really happy with that balance. It's a little less that now, and it might be a result of COVID. It might be, you know, that the honeymoon period is over or whatever. It might be the definition of what makes me excited and happy has also changed. It might not be anything that is external, right? And so I've told, you know, I guess my mentees, you know, as they're starting faculty positions as, you know, assistant researchers to assistant professors, to think about that as a really important goal in your life, to, you know, that 60, 70% of the time you're doing things that are exciting to you, and that will keep your life happy. I think that's a key lesson, you know, for us at this stage. I think, you know, mid-career is tough because you can be really hard on yourself. You can look back at every single possible failure, right? And you can leave those as markers. And I'm not going to sit here and list mine because I can cry about it. I'll probably start crying. But they can actually take up a whole lot of space in your head. Like, I didn't accomplish this, or I didn't, and that, and one of the really tough things is comparing yourself to other people, right? And so, to some extent, I look at LinkedIn less and less nowadays because everybody's posting their career achievements, but nobody ever posts career failures on LinkedIn, right? And you start to get into a mindset where you feel like you fail more often than other people. And it's really important to avoid that and to kind of focus in on what you've been good at and what you've done well, and not recognize those, not give extreme voice or over importance to things that you deem as failures, and and turning those failures into positives as well. So that, I think, is, and I struggle with that all the time, but I think that is a huge, figuring that piece out is a good piece of advice for us at mid-career. For early career people, one of the things that I tell them is imposter syndrome. Just avoid imposter syndrome like crazy, because I always felt that way, like I didn't belong in this room, or I wasn't smart enough to be here, or or whatever. And it's really it's such a wet blanket on success, and so that's a key thing to avoid. And you should listen to.
And it hinders your ability to learn and your ability to teach.
Exactly.
If you think, I struggled so much with imposter syndrome when I first started out professionally, because I didn't get my PhD. And so I went straight into the chemistry of dyes, pretty much from NIH, and then I remember it was actually the same week I gave a talk at Hopkins, and I gave a talk in front of Mario for the first time. And I had heard about how hard he was to be. And after I left, I was like, you know what, Kelly? I'm not positive you are an imposter.
Yeah,
like, exactly. I think that you should just wait until someone tells you you are.
Yep.
And then maybe do something about it. But until then, don't make the assumption that that's the truth, because I don't think it is.
And even if they tell you that, so what's the language requirement here? I mean, do I have to keep it clear?
No, we're not PG-13. We're fine.
Okay, fuck them, right? And so you take constructive, you have to recognize, because there's criticism all throughout science, right? I mean, if there's one thing that's a constant in science, it's skepticism and criticism. I mean, everything we do is about that, right? Peer review is that, every paper comes back with that. Everybody's evaluating your proposals, and so there's constant criticism surrounding you in your life. And you have to recognize that a lot of that is, you have to address it, you have to deal with it, and, and grow from it. But, and
then let it go.
Yeah, exactly. What's to say that person knows better than you,
Sure.
And so he's just another idiot like you, so not like you, but, like, that's what you should tell yourself. So,
very good advice. I like that very much.
So, anyway.
I think we're probably at our time limit. What do you think, David?
Yeah, that sounds about right.
Yeah.
Man, thank you so much for joining us. I love talking
guys. This was a whole lot of fun, and I wish you a lot of luck as you're developing more of these podcasts, and I'm looking forward to learning about other people in the community as well, and topics.
Absolutely.
Awesome.
Well, thank you.
Thank you so much.
Yeah.
And to our listeners, thank you so much. It gets lonely on the YouTube, so leave us a comment or come and visit us at ChuG Cytometry.com and chat with us. We want to hear from you. We want to hear if you enjoy the podcast, and if you have any topics you'd like us to discuss. Talk to you guys later. Thank you so much.
Absolutely.